All right, students, a kid asked me to go ahead and make a video explaining stuff in this quiz we took. So let's do that. Let's go through it. Okay, so in this question, it said, what's happening in step A of bacterial binary fission? So here we see a bacteria in step A going to step D, where it's two bacteria. Which of the following goes to step A? That would be the chromosome getting duplicated or replicated. And you can see here, this is showing you that chromosome turning into two. Here you see the chromosome separating more, the cells elongating. And here you're getting a bifurcation in the middle, starting to separate these two cells. And the chromosomes are completely separated at that point. And then finally, in step D, that bifurcation is complete, and we have two cells where this cell has made a membrane in between them, along with a cell wall. So notice that there are two lines making this barrier of the cell. There's an inner line and an outer line. And that's one way of showing both the membrane and outer cell wall of the bacteria. Bacteria can have a cell wall, unlike eukaryotes that are in the animal kingdom that don't have a cell wall. So here's a cell membrane and a cell wall. So the next couple questions we're dealing with some of the things that I just mentioned. Step B, chromosomes are separating. That would be that answer. Step C, chromosomes have separated. Cell membrane and cell wall is forming between the two cells. So here's is forming. And then we get to complete separation, which the cells are separating where there was one, now there are two. That would be step D. All right, moving on. Yada, yada, yada. What kind of plates are these? These are blood agar plates, or blood agar petri dishes. So the red's coming from sheep blood. We watched a 2-minute video on the exact video that this picture was pulled from. And we have different colonies here. These are likely bacterial colonies. If you wanted to make sure that those were indeed bacterial colonies, you could put an antifungal chemical like they treat for, what do you call it? Foot fungus. So tofactin, tinactin, or something like that. You could actually spray into your agar while you pour it and make this an antifungal plate, kind of guaranteeing that these are going to likely be bacteria and not fungus. But these are bacterial colonies, as they explained in the video. So what types of plates are those? They're blood agar. In the video, they said it was 5% sheep blood. And continuing on, some of the nasty bacterial colonies that were grown on the petri dishes. So Nigeria is a country. Niglaria, I think, was one thing that was mentioned in the video. But I purposely threw this in here to throw you off. And either way, the nasty bacteria that kind of were similar to these names in the video were pneumoniae, not bacillus. And the particular one that this person talked about for a bit in the video was Staphylococcus. Staphylococcus pneumoniae, to be exact. So that's the answer there. Some other things that the scientists talked about in the video were methicillin-resistant Staphylococcus bacteria, which can get its antibiotic resistance from being transformed with a plasmid. And that's related to something else that we had learned about a couple weeks ago too. The Bacillus anthracis, that's a biological weapon that was kind of famous in the late 90s, early 2000s, which has nothing to do with this video and wasn't even mentioned in it. Some of the nasty bacterial colonies that were grown on the plates were bacteria found in poop called fecal bacteria. And that was one of the interesting things that the scientists talked about. I think it was 2 or 4 of the cell phones that were swabbed out of the total 28 were fecal bacteria, meaning those people probably took those cell phones to the toilet with them. And the scientists talked about that. These are just, had you paid attention in the video, you would have known that there is no discussion whatsoever of earwax or professional sheep herders. You would have, yeah. So continuing on. Let's see. Are all of the bacterial colonies the same size? Well, that's a colony, that's a colony, that's a colony, that's a colony, and you can see that they're all not the same size. So no, they're not the same size. Why are they not the same size? Well, we sort of discussed this in our laboratory experiment that we did with growing our own plates and bacteria. So yeah, different species of bacteria can have different growth rates on different foods. Yeah, that's it. All the other things are just, non-answers really. How are these bacterial plates similar to the ones you're growing in class? They both contained agar and have been swabbed or inoculated. That's the answer there. Continuing on. How are these bacteria? So this should have been, meant to delete the word how there. And it was meant to just say, are these bacteria on these plates microbes? And the answer is yes. And I don't want to say that that's a trick question, but if you remember from discussing, microbes are things you can't see with your eyes. Or I should say microorganisms that you can't see with your eyes. But you can see these colonies with your eyes. So how are those microbes if you can see the colonies with your eyes? And the answer is you're not seeing one organism, you're seeing millions. Continuing on, why can you see these bacteria with your eye if they're microscopic? And I just told you that you're not seeing one, you're seeing millions. Are all of these plates, are all the bacteria on these plates pathogens? And the answer is absolutely not. The scientist said that in the video and we discussed it in class. So no, they don't all cause disease. They're not all pathogenic. What's a pathogen again? It's an organism that causes disease in another organism. It isn't an organism that has pathology. It's not an organism that is diseased. Microbes are organisms that can be seen with a microscope. And flea, ticks, and mosquitoes are parasites. They're not really pathogenic, but they can give you pathogenic organisms. Staph aureus is a pathogen, though, and not all fungi are. Okay, continuing on. Are all microbes pathogens? Nope, totally not. As a matter of fact, some of the bacteria on this plate are probably things that you could accidentally consume and you'd just be fine. I did say some of them though. So obviously, big problem if you do that because you probably get some contamination and get sick anyway. All right. Examples of pathogen that you don't need a microscope to see would be tapeworms and hookworms. So you can see those critters. They are not microbes, but they are pathogenic in humans and other animals. Fungus are not necessarily pathogenic. So that one's ruled out because of that. Not all bacteria are pathogens. And malaria you cannot see without a microscope. Okay. Why does the DNA need to replicate in the bacterial cells? Because if it didn't, one of these cells will be missing some DNA. And that answer is written down below there. Let's see. This one. Continuing on, if the parent cell's chromosomes don't separate, what will happen? The cell will package the DNA into a separate nucleus. That doesn't sound right. The cell will either not divide or one cell will get twice as much DNA, meaning having 2 chromosomes. That would be the answer there. Continuing on. Why is Mr. B giving me a quiz on yesterday's materials where most of the answers are kind of given to us? Let's see. Because they're still not the easiest questions in the world, however, how many people discuss this topic on a daily basis? Let's see, what else did I have written there? Oh, how many people discuss this on a daily basis? Well, this answer sounds right, but it's not the best answer. How about because a great part of learning is discussion, and your teacher discusses things in a variety of formats. That sounds like a good answer. Continuing on, the fecal matter question. So yeah, we had Klebsiella pneumoniae, not Klebsiella bacillus. And what is similar in this picture to Mr. Avery's experiment that we learned about 2 days ago regarding RNS bacteria? It's referencing bacteria that cause pneumoniae. So in Avery's experiment, they discussed Streptococcus pneumoniae, which caused pneumonia and killed the mice when we had smooth bacteria. And in this case, this is referencing other bacteria that cause pneumonia. And 4 of the phones that were swabbed had poo-poo bacteria on it, otherwise known as fecal bacteria. Continuing on, if the mouse survives, here's Avery's experiment. If the mouse survives, it means it was injected with not smooth staph, because live smooth Staphylococcus pneumoniae killed the mouse. Rough staph pneumoniae that was added to heat killed smooth staph bacteria would kill the mouse. Heat killed smooth did not kill the mouse, and saffron is obviously not a right answer. So it's one of these 3, and I already forgot the question. What was the question again? If the mouse survives, that means it was treated with heat killed smooth staph. Smooth staph, when you heat killed it, did not kill the mouse. Okay. How does rough bacteria turn into smooth bacteria? Dead smooth bacteria give their DNA, usually as plasmids, to the live rough bacteria in the process called transformation. Transformation can lead to bacteria being transformed by antibiotic resistance plasmids from other bacteria. And that looks like that's the end of that. So let's go ahead and stop this video. See how do I stop recording?